So now we're arriving at one of my favorite parts of our biochemistry tour, which is the point where we get to start talking about the four major categories of biomolecules or macromolecules that are the big players in the cell functions that we're about to explore. So those four major categories are as follows. There's the carbohydrates, the lipids, the proteins, and the nucleic acids. So in this video, I'm going to talk generally about the polymers overall, so what we need to know about these molecules. And then for the rest of this video, we're just going to focus on the first macromolecule group, the carbohydrates. I've subdivided the remaining macromolecules into individual videos so you can really take your time with them. Because if a lot of this information is new to you, you may find that you want to spend a lot of time on these topics, and it's easier to do that if the information's broken up into smaller pieces. I'll also note that while it's extremely useful to practice drawing these out in order to understand them, I am not going to ask you to draw them out on an exam at this stage. So, what is it that we need to know about these molecules to have a good working understanding of cell function? So first off, we need to know what they are. So most of the substances we're going to talk about are found in cells as polymers. And so polymer is just a term for a molecule that's composed of a bunch of repeating units. So we can start to understand how these molecules function by considering the types of building blocks they're made from. And what we want to understand has two sides. First, how are they constructed? So their construction can give us some insights into how they function in cells. But along with knowing how they're constructed, there's kind of the inverse of that, which is how do they vary? So it's that variation that creates a possibility for a wide range of uses for each type of polymer. And in some cases, we're also going to encounter some special subcategories within a given type of polymer. And we're going to cross that bridge when we come to it. So once we know something about those individual building blocks, we're then going to want to examine how those building blocks get organized. So how are they chained together, and what are the consequences of that? And along with that, we're also going to want to start talking about where they're found inside of cells. And that's going to give us some ideas as to the specific functions of these macromolecules inside of cells. And again, make note that the functions of a polymer are very closely related to the properties of that polymer. And lastly, there's another question for us to consider, which is, well, why do we need to know these things? And right here now, the biggest reason is that if we understand how these polymers are constructed, where they're found, and what they do, we're going to be better prepared to understand the next layer of complexity that we're going to add to our understanding of living things, about how cells work. And this is really going to become clear as we continue to progress and start putting these pieces together with larger scale cellular function. Okay, and so without further ado, let's dive in and take a look at the carbohydrates. Okay, so carbohydrates are built from individual units that are sugars, also known as monosaccharides. But there are a lot of different kinds of monosaccharides possible, but one thing that they generally share in common is that they're composed of some multiple of the unit CH2O. So say CH2O times X. Okay? So the most common monosaccharide, for example, is glucose. And that has a molecular formula of C6H12O6. Okay? The other trademarks of sugars are the presence of a carbonyl group, right? So the carbonyl group is a C double bonded to an O, and then a bunch of hydroxyl groups. So we're going to look for a lot of OHs or hydroxyl groups. There's a couple different ways that the simple sugars get depicted. So we're going to start out by drawing out the linear form of glucose. Here's the linear form of glucose. So we've got 6 carbons, and then up here, up top, this carbon's bonded to a hydrogen, and then it's got that carbonyl group as well. So imagine I spelled that right. Okay, then here we've got hydrogen and a hydroxyl. We've got a hydroxyl sticking out on the other side. And then for these last 3 carbons in this chain, we've got hydrogens on the left, and we've got 3 hydroxyls on the right. Okay. One thing with these molecules is, so we've got a whole bunch of carbons. We need to keep track of these 6 carbons. So we're going to number the carbons, and we're going to start the numbering with the carbon that has this carbonyl group hanging off of it. So here's 1, 2, 3, 4, 5, and 6. And just for fun, I'm going to show you an enantiomer of glucose called galactose. So let's draw galactose out right here. So you've got 6 carbons. We've got our carbonyl up here. And, drum roll please. So the difference here, so here's galactose. Right, so we're spotting the difference. Same kind of numbering system is going to apply. But what you'll notice here is at carbon number 4, the hydroxyl is in the opposite position from on glucose, right? So here's 1, 2, 3, 4, 5, 6. That's an enantiomer of glucose. Even though this might seem like it's a really trivial difference between these molecules, that can actually have major implications for how these molecules wind up getting used. So now let's take a look at another 6-carbon sugar. So next 6-carbon sugar here is fructose. So there's my carbons. And in this case, the carbonyl group is found on carbon number 2, right? Right, so there's our big difference there. So again, we're going to numbering these carbons. So same molecular formula, but different structure. Okay. So now I want you to take a second to think, what kind of isomers are glucose and fructose? And I'd suggest that you pause the video, think about this, and write down your answer. And I'm going to be sneaky, and I'm not actually going to tell you what the answer is here. So now carrying on, so this linear form of glucose is actually pretty rare. And instead, most carbohydrates are found in a ring structure instead, because a ring structure is a lower energy form, and things tend to move towards a lower energy form. So now we're going to walk through how we get to the ring form from the linear form. And I'm going to start out drawing carbon number 1 down towards the bottom here. Here's carbon 2, 3, 4, 5, and 6. And for carbon number 6, I'm going to just abbreviate all of this stuff by calling this carbon CH2OH. And then on carbon number 5, so carbon number 5 has a hydroxyl and a hydrogen hanging off of it. So I'm going to draw the hydroxyl over here. And then I'm going to draw that hydrogen down here. And then carbon number 4 has got both hydrogen and hydroxyl. Same for carbon 3, carbon 2, and then carbon 1 has got that carboxyl, right? Okay, like so. So let's just renumber these carbons so we can keep track of them again. So here is 1, 2, 3, 4, 5, and 6. Okay. Basically, so this is going to convert to the ring form by an interaction between this hydroxyl group and this oxygen double bond right here, okay? So what's going to happen is this hydrogen is going to get transferred to this oxygen. And in the meantime, this oxygen is going to bind to carbon, okay? Okay, so I'm going to draw that out here. So if I'm transferring this double bond, so that's now a hydroxyl, and I've transferred that hydrogen, and now there's going to be a bond here. Okay, and so now I've gone to carbon's ring form. And one other thing I want you to notice is before I did that, this attachment point here between carbon 1 and carbon 2, right, that's a single bond. So what that means is that it's possible for this hydroxyl and this hydrogen to be in the opposite orientation, right? Because with a single bond here, before we connected up, this whole unit could potentially rotate in space. And so it's possible that we wind up with this configuration I just showed you, where the two neighboring hydroxyl groups are next to each other, or it's possible to wind up with the opposite configuration. Where those hydroxyl groups are opposite from each other. And when they're next to each other, that gives us a form of glucose that's called alpha glucose. And in contrast, if these two hydroxyl groups are opposite from each other, that's going to give us what's called beta glucose. Okay. And just like I showed you with the differences between glucose and fructose, this difference between alpha glucose and beta glucose can make a huge difference when it comes to how other molecules can interact with these glucoses. We're going to see more about that in just a moment. And just to walk through this concept again, I'm going to speed through a second example of ring formation for fructose. So redraw fructose. And in this case, okay, so in this case, here's my carbonyl group. Okay, so here's what I start off with. And in this case, here's carbon 1, right up here. 2, 3, 4, 5, 6. It's carbon 1, 2, 3, 4, 5, 6. Oops, and I left a hydrogen off of carbon 5. Okay, so in this case, what winds up happening is a transfer of this hydroxyl group onto carbon number 2. And so then what we'll wind up with is carbon number 5. Oops. Sorry, that hydroxyl group there. Carbon number 5 bonded to that oxygen, and then hydroxyl group coming off of carbon number 2. So that's a slightly more complex reaction. Again, you don't need to dwell on the details, but just be aware that there are multiple ways that rings can form. Again, we're not going to go into a ton of detail here, but just to start to get you familiarized with these, I'm also going to draw out 2 other sugar molecules that are going to reappear later. Okay, and let's number these. So we're going to number starting from this carbon. Okay, and then notice that the only difference between these two carbons, and these are 5 carbon sugars, is right here at carbon number 2. So these two 5 carbon sugars are collectively known as deoxyribose, right? Deoxy because there's no oxygen here. And then this other 5 carbon sugar is then just called ribose. And again, those are going to reappear later on in this course. Glucose and fructose, which I showed you earlier, are referred to as the hexoses because they are 6 carbon sugars. But it's also possible to have just 5 carbons and be a pentose. And again, just notice that these two 5 carbon sugars are very similar to each other. Okay, so that's what the fundamental building blocks look like for carbohydrates. But the next question is then, how do these fundamental building blocks connect up to each other to form larger molecules? So we're going to look at an example of this. And the specific example we're going to do is we're going to combine a glucose molecule with a fructose molecule. So again, here's fructose, and here's glucose. And again, to keep ourselves oriented, carbon number 1 on glucose is here. And carbon number 1 on fructose is up here. Okay? Okay, so in doing this, so biologists like to give things fancy names. So instead of just calling a sugar, you know, they're saccharides. So if it's just one sugar, then it's a monosaccharide. But if you combine 2 together, then it's going to be called a disaccharide. Okay? Thankfully, after that point, we basically just switch over to the prefix that means many. So beyond that, we'll be talking about polysaccharides. Anyway, so for making a disaccharide, the way that these combine to form a disaccharide is these 2 hydroxyl groups are going to interact with each other. And so it's the hydroxyl groups hanging off of carbon number 1 and carbon number 2 on fructose. And so under the right circumstances, with a little bit of help from some friends, these 2 hydroxyls get combined and a molecule of water gets released. So that's going to leave the scene here. And when that happens, what we're left with is an oxygen holding these 2 together. And the connection position here is named according to the 2 carbons that get joined together. So you've got carbon 1 and carbon 2. So this winds up being referred to as a 1,2 glycosidic linkage. And again, you know, don't get too caught up in this name. Just be aware that you know, this the position of this connection can vary, and so it's named according to where that position happens. And so now the result here of combining together glucose and fructose is a disaccharide that is known as sucrose, aka table sugar. And I think once you've seen one disaccharide, I hope it's a relatively small conceptual leap to start to imagine bigger polymers forming, right? So we've got a whole bunch of hydroxyl groups all over the place here. So there's a lot of potential business points for these carbohydrates to be connected to each other. And so, for example, I'm just going to show you what it looks like if we string together a whole bunch of alpha glucose molecules. And in this case, I'm going to simplify the drawing to some extent. I'm going to omit the hydrogens, and instead of drawing each individual carbon with a C, I'm going to indicate it by the intersection point of lines. So you'll see the oxygen, but then at every other point here, there's just a line, and then up here I will continue to put CH2OH, okay? And we've got hydroxyl there. Okay, and you can imagine that I could keep going off of either direction here. So a chain of alpha glucoses connected together, and this is right here's carbon number 1, and here's carbon number 4. So this is a 1,4 linkage. So this whole chain has its own name, and the name for this whole chain is starch. Okay, and so that's alpha glucose, right? So let's take a look at what we get if we string together a bunch of beta glucose molecules. And the same sort of concept applies. We can keep building on this chain. So this again is a 1,4 linkage, right? So carbon number 1 is adjacent to oxygen, and then carbon number 4. So we've got 1, 2, 3, 4. So there's carbon number 4. So it's another 1,4 linkage, but with beta glucose. And so in this case, you'll notice that this end group is flipped every other, right? And so this configuration is called cellulose. And I'm going to show you another picture from your textbook that highlights some other possibilities for these polymers. Okay, so this picture brings us to the next things to know about carbohydrates, which is where they're found and what kinds of functions they have within cells. So let's talk about functions, and that's going to give us a clue as to where they might be found. So first, a lot of carbohydrates are used to create structure for cells. So for example, cellulose and related molecules create structure for plant cells. And so plant cell walls that surround the outside of a plant cell contain a whole lot of cellulose. Okay, animals also use polysaccharides to provide structure, typically in the form of chitin. And chitin is also built out of those long chains of polysaccharides, and then there's usually some other modifications thrown in. So they're also found in extracellular spaces. And extra means outside of the cell. So the second major function of polysaccharides is that they serve as a form of energy storage. So plants tend to store glucose molecules in long chains as starch. And starch has occasional branches, and then animals store glucose molecules in a more branched form known as glycogen. And these are both stored in specific locations inside of cells. And we're going to talk more about that when we talk about cell structures pretty soon. So let's go back and look at these pictures of polymer possibilities. Okay, so up top here is an unbranched chain of glucose. So this is a storage molecule called amylose. No need to remember that name. Starch will also contain some partially branched chains of glucose, such as the amylopectin that's pictured here, but they're largely straight chain. And then here's glycogen in contrast, so it's got a whole lot of branching structures to it. So found in this person's muscle cell illustrated here. And then in contrast, here's a whole set of cellulose molecules. So these are very much straight chain, and adjacent chains of cellulose are held together with hydrogen bonds in between them. Okay, and I'm going to wrap up this section on carbohydrates by noting that there's some trade-offs to storing energy in the bonds that are found in carbohydrates, and that these molecules are fairly bulky. So there's limits to how much carbohydrate can fit inside of a given cell. So for something like, one of these potatoes, that's not going to be a huge issue because it's just going to make the potato bigger. But for something like you or me, that can start to turn into a problem because we have to carry everything around with us as we move around in the environment. So for our next type of polymer, we're going to see one potential solution to this storage dilemma.